Welcome back to another video by Dr. Pembroke. Today's topic is hepatitis B, which affects one of the human's favorite organ, the liver. The liver lives in the upper right side of the abdomen and is the largest internal organ. It can convert carbohydrates into glucose for instant energy or into glycogen for storage. It also produces transaminase enzymes to break down amino acids and bile to break down dietary fats. It also filters medications and drugs to neutralize or convert them into other forms. Toxic byproducts like ammonia are also turned into urea for excretion. Hepatitis B is the inflammation of the liver by viral infection of hepatitis B virus. The most common way of spreading HBV is when HBV-infected blood or body fluids enters the body of a non-infected person. From unprotected sex, needle stick injury, sharing personal items, and from mother to baby during birth. It is not spread by food or water or casual and social contacts. When HBV virions enter hepatocytes, genomic HBV DNA is transported to the nucleus, where circular DNA is converted to cccDNA, which becomes the template for RNA transcription. Hep B proteins are produced via reverse transcription and translation. E antigens and viral DNA are also secreted into cells. Hep B is usually diagnosed when HBV DNA, surface antigen, and E antigen are detected in serum tests after exposure. E antigen correlates to the level of HBV replication. Many people with Hep B have no symptoms and don't realize they had an infection, but some people may experience some of the following symptoms. In acute infection, there is an incubation period of 45 to 180 days where clinical symptoms are not detected. The disease onset occurs several weeks after HBV elimination. As cells are infected, MHC1 molecules will attract virus-specific CD8, CD4, and cytotoxic T cells to infected cells, leading to cytotoxic killing and apoptosis of hepatocytes, hence the symptoms of inflammation of liver. When hepatocytes are damaged, amino transaminase enzymes are leaked into bloodstream. The increased level of ALT and AST indicates the degree of hepatocellular damages and T-cell-mediated liver injury. By the time of peak ALT level, most of HBV DNA in serum and liver are usually cleared. Jaundice may also appear as a symptom due to conjugated bilirubin in urine causing dark urine. When hepatocytes lining the bile duct apoptose, CB is leaked into the blood and to the kidney where it is filtered into urine. IgM and IgG antibodies are also produced to attack core antigen and surface antigen respectively, shortly before the onset of clinical symptoms. This indicates initiation of recovery and also diagnosis for Hep B. Chronic patients have similar initial serological marker patterns, but CD4 and CD8 response are insufficient, therefore viral replication persists. There is an estimated of 257 million people infected with Hep B in 2015 worldwide. Hep B infection in African, Southeast Asia, and European regions have a higher rate. In Australia, an estimated of 232,600 people live with chronic hepatitis B. A third of Australians are not diagnosed and 2/3 were born overseas in endemic countries or identified as Aboriginal Torres Strait Islanders. Prevalence of Hep B infection also increases for people born in high prevalence areas or people in higher risk groups. There was a total of 6,502 newly diagnosed cases of hepatitis B infection in Australia in 2015. The Hep B rates are higher amongst males than females and 3 times higher in Aboriginal population than non-Indigenous population. Northern Territory also has the highest rate of Hep B infection in Australia. However, due to the impact of vaccination programs, new hepatitis B infection has declined in Australia in younger age groups.